learners. I am Dr. Maneesha Pandey, Assistant Professor, Biochemistry Discipline School of Sciences, Indira Gandhi National Open University. In this session, I am going to discuss with you about nucleic acids. This is the part of our course Molecules of Life BBCCT 101 under the BSc Biochemistry Honours Program. When we listen about nucleic acids, what comes to your mind? What is a nucleic acid? Why it is so important? Well, nucleic acids are polymers of nucleotides. They are one of the important macromolecules, biomolecules you can say, and the nucleic acids have been classified into two types, DNA and RNA. DNA stands for deoxyribonucleic acid and it is found in the nucleus of a cell. Now, small amount of DNA is also found in mitochondria and chloroplasts. They are the extra nuclear organelles, and that is why the DNA present in mitochondria and chloroplast is termed as extra chromosomal DNA. Ribonucleic acid or RNA is found throughout the cell. Now the question arises, why nucleic acid is so important? Well, nucleic acid is important because it is considered and it has been proved to be the genetic material. So, it is the information storage molecule. We can say that nucleic acid carry genetic information from one generation to another generation. And this ability to store and transmit genetic information from one generation to next generation is the fundamental condition of our life also. Now, let us look how this flow of information takes place. Flow of information is also known as central dogma of molecular biology. Central dogma means something which is very important. How this information flow takes place? If you look at the figure, you will see that the message encrypted in the DNA double helix is getting converted into RNA, and then this RNA is further getting converted into protein. The process of conversion of the message encrypted in DNA or coded in DNA and its conversion into RNA is known as transcription, and then the encrypted message from RNA is getting converted into protein, and this process is known as translation. Now, this is a unidirectional flow of information from DNA to RNA to protein, and this flow of information is very vital phenomenon. If you look at another picture, you will see that here the DNA is localized within the nuclei of the cell, and then when it is getting converted into RNA, especially messenger RNA, it is coming out of the cell where ribosomes attach to it and convert it into protein. Now, the synthesis of functional biological product, whether it is a protein or RNA, whenever it is required, a segment of DNA molecule that contains this information undergoes synthesis, and this piece or segment of DNA coding for a particular functional biological product is termed as gene. Now question arises, if this message is so important, how the information is getting stored? Information can be stored by any object which possesses more than one distinguishable state. For example, if you look at beads, if it is a small bead or a bigger bead, it may convey different messages. Then colors of bead symbolizing different things. Similarly, in DNA, the information is stored in linear sequences, and this particular sequence of 4 different elements along its length, which are known as nitrogenous bases, are storing information here. We will talk about nitrogenous bases further. The structural arrangement of these 4 different bases along its length and interaction make DNA structure so well suited that duplicated information storage permits repair of damaged information as well as a unified mechanism of replication. If information is stored in DNA, then what is the significance of RNA? When we were talking about central dogma of molecular biology, we saw it is acting as a intermediate between DNA and protein. So one of the cellular uses of RNA is as a temporary information carrier. RNA ordinarily does not participate in replication or repair activities, but in addition to handling information, some type of RNA molecules have also been found to have structural or catalytic activities, and this ability of RNA to perform all these roles has led to the belief that in evolution of life, RNA appeared much before DNA or protein. So, let us look at the structural aspect of DNA. The building block of nucleic acids, as I said, and you all know also, are nucleotides. So, nucleic acids are very well termed as polynucleotides or polymer of nucleotides. RNA or ribonucleic acid is a single nucleotide chain, whereas DNA consists of two nucleotide chains and these chains are linked together. Now, let us look at the structure of a nucleotide. So, whereas DNA consists of two nucleotide chains which are linked together. So, let us look at the structure of a nucleotide. If you look at the figure, you will notice three things. There is a phosphate group, there is a sugar molecule and there is a nitrogenous base. Now, to a pentose sugar, at one end phosphate group is attached, at another end the nitrogenous bases are attached. So, nucleotides are composed of three things: nitrogenous bases, sugar and phosphate, and a nucleotide can have 1, 2 or 3 phosphate group links together. When phosphate group is not attached with the nucleotide, it is termed as nucleoside. So, nitrogenous bases along with sugar make nucleoside, and when phosphate get attached to it, then it becomes a nucleotide. I just mentioned that the sugar in nucleotide is a pentose sugar. You can question why it is not a hexose sugar or some other sugar. Experimentally, it has been found that because of steric hindrance, other sugars were not suitable to fit, and it led to unstable structure formation with other compounds of nucleic acids. So, pentoses are the best suited for the formation of the nucleic acid structure. And here you must remember one thing, that whenever we are numbering the carbon of a pentose in a nucleotide or a nucleic acid chain, they are assigned prime designation just to distinguish them from the numbered atoms of the nitrogenous bases. Now, if you look at the figure, you will notice two things. There is a deoxyribose sugar in DNA and a ribose sugar in RNA. So, what is the difference between deoxyribose and ribose? If you see at the 2' position, in ribose sugar, additional hydroxyl group is present, while in deoxyribose, hydroxyl group is not present or a hydroxyl group is missing. This additional hydroxyl group in ribose makes it prone, you can say, or other way round, due to the presence of deoxyribose sugar, which is containing one less hydroxyl group in DNA. DNA becomes more stable than RNA, and this is why it is very useful for keeping the genetic information safe and serving as a storage molecule. Now, we will talk about nitrogenous bases. Nitrogenous bases were first identified in late 1880s by Emil Fischer. The nitrogenous bases are derivatives of two parent compounds, purines and pyrimidines. Both are heterocyclic compounds made up of carbon and nitrogen, and both have got the planar structure. Planar means they are like flat molecules. So, both are heterocyclic and both are planar in structure. Now, if you look at the structure, if you will see, purine has got double ring and the pyrimidine has got single ring. And 5 commonly occurring nitrogenous bases in DNA are cytosine, thymine, uracil, which are pyrimidines, single ring structures, and adenine and guanine, which are double ring structures and purine derivatives. Now, if you look at the numbering, you will see that the purines are numbered in anti-clockwise direction, starting from N1 to N9, while the pyrimidines are numbered in clockwise direction. Now, we will see which base is present in DNA and which is present in RNA. The major purines of both DNA and RNA are adenine and guanine. So, purine is common to both DNA and RNA. From pyrimidines, cytosine and thymine occurs in DNA and cytosine and uracil occurs in RNA. So, you can say thymine of DNA is replaced by uracil in RNA. Now, in addition to these 5 nitrogenous bases, some unusual bases are also found in nature, for example, hypoxanthine, xanthine, uric acid. And in RNA, unusual bases like 5-methylcytosine, pseudouracil, N6-methyladenine also occur, and you will see it later. Now, let us look at the entire component of nucleic acids together. If you look at the structure, you will see that from amongst the nitrogenous bases, thymine occurs only in DNA, uracil occurs only in RNA, while adenine, guanine and cytosine are common to both DNA and RNA. Phosphate is commonly found in DNA as well as RNA structure. Amongst the sugar part, 2-deoxyribose is present in DNA while ribose is present in RNA. So, all the 3 things together, when they join, they make the structure of nucleotide. Let us look at the structure of a nucleotide. How the phosphate is joined? We just talked also. The phosphate is joined with the 5th carbon of the pentose sugar, and the nitrogenous base is attached at the 1st carbon of the pentose sugar. Now, the N9 of purine or N1 of pyrimidine is actually getting bonded with the C1 of the pentose in a N-glycosidic bond formation. So, when the N-glycosidic bond formation is taking place, what is happening? A molecule of water is eliminated and the N9 or N1 of the nitrogenous base gets linked with the C1 carbon of the sugar. This N-beta glycosyl bond is formed by the removal of an element of water, and the hydroxyl group comes from the pentose while the hydrogen comes from the base, which is very much like the O-glycosidic bond formation. Now, the entire structure of nucleotide you can visualize. Now, we will look at the IUPAC nomenclature. I was just mentioning adenine, guanine, cytosine, thymine or uracil. They are the common names, and one letter symbol has also been assigned to it, adenine being A, guanine being G, cytosine C, uracil U, thymine T. But to have a uniform system of nomenclature, IUPAC naming has been done, and based on the chemical structure, adenine is known as 6-aminopurine because at the 6th position amino group is present in the purine ring. So, this is known as 6-aminopurine. Similarly, guanine is known as 2-amino-6-oxypurine, while cytosine is 2-oxy-4-aminopyrimidine and so on. So, they are named according to the basis of the functional group present at definite positions. Similarly, the nucleoside and nucleotide nomenclature is also done. When adenine is a base in a nucleoside where sugar and base are joined together, it is termed as adenosine in RNA, and in DNA it is termed as deoxyadenosine because of the presence of deoxyribose sugar. Guanine becomes guanosine or deoxyguanosine, and corresponding nucleotide is termed as adenylic acid or deoxyadenylic acid. Now, we will talk about the pyrimidine bases. When they are present, cytosine is termed as cytidine or deoxycytidine, uracil is termed as uridine, and thymine is termed as deoxythymidine. Here, we are not saying thymidine and deoxy or uridine and deoxy because they are present specially only in DNA and RNA. Here you must have noticed that the names of purine nucleosides end in ocene, while the name of pyrimidine nucleosides end in idine. Now we will talk about nucleotide linkage. So far we were discussing the composition of nucleotide, that how the different components come together and form the structure of a nucleotide. Now We know that nucleic acid is a linear polymer of nucleotides. So how the nucleotides are linked together? The successive nucleotides of both DNA and RNA are covalently linked through phosphate group bridges or phosphodiester bonds, and what is happening here is a 5' phosphate group of one nucleotide unit is getting linked to the 3' hydroxyl group of the next nucleotide. So if you look at the entire structure, what is happening is a nucleotide comes and joins to another nucleotide, and the 3' hydroxyl attaches with the 5' phosphate of the another nucleotide, and this creates a phosphodiester linkage. It's an ester linkage, ROR linkage, and this bond formation links the two nucleotides. Simultaneously and successively, the nucleotides come and attached with the 3' hydroxyl group of next nucleotide. So what is the significance of this phosphodiester linkage? Significance is, one, we are getting a linear chain, a linear polymer chain is created. Then all the phosphodiester linkages have the common orientation or same orientation along the chain. This gives linear nucleic acid strand a specific polarity and distinct 5', 3' end. Means at one 5' end, if one nucleotide is present, then another will come and join here. So this is how the chain is moving from 5' to 3' end. So this is providing a specific polarity as well as a distinct 5' and 3' end. And theoretically, you can see that the 5' end of a nucleotide will lack nucleotide at 5' position, while the 3' end of the nucleotide will lack another nucleotide at the 3' position. What this means? This means the 5' end of a chain will be vacant and the 3' position of the chain will be vacant. This phosphodiester linkage is creating a single linear chain, but we know that DNA is a double helix. So how the double helical structure is formed? Well, here we will talk about base pairing. If you look at the structure, you can see that the thymine has bonded with adenine, the guanine has bonded with the cytosine. The dotted lines represent hydrogen bond, and there is two dotted line, or you can say two hydrogen bonds are present between thymine and adenine. 3 hydrogen bonds are present between guanine and cytosine. Similarly, uracil and adenine also form 2 hydrogen bonds. So, two different nucleotide chains are getting joined together with the help of hydrogen bonds, and this is known as base pairing. We all know that hydrogen bonds can form whenever a hydrogen atom can get shared by a donor such as amino group and an acceptor such as a carbonyl group. These hydrogen bonds formed between the bases of DNA are strong because in all the cases, 3 atoms participating in hydrogen bond formation lie almost nearly in straight lines. Here you must have noticed that purines and pyrimidines are different in shape and sizes. However, the pair of these molecules like adenine thymine or guanine cytosine do possess regular shapes. And not only these pairs are regular, but they are further stabilized by the hydrogen bonds. Base pairing, that is hydrogen bond formation between a large purine like adenine or guanine on one chain and a smaller pyrimidine, that is thymine or cytosine, of which each is having a single ring on the other chain, occurs effectively without any steric hindrance. So this base pairing is creating a double helix or joining the two helix together. And this basic structure of DNA of double helical nature was deduced by Watson and Crick using 3 pieces of information: X-ray diffraction data, structure of the nitrogenous bases, and Chargaff's finding, that is base pairing. If we talk about the salient features of Watson and Crick model of DNA, which was proposed in 1953, it has some important features. It says the 2 linear nucleotide chains run in opposite direction, that is anti-parallel orientation. If one chain is moving up, the another chain is coming down. So these two oppositely running chains are coiled around a common axis to give rise to a helix. Then the sugar and phosphate backbones are on the outer side, while the purines and pyrimidines bases lie on the inner side of the helix and are nearly perpendicular to the axis. What this means? If this is forming the sugar and phosphate backbone, then it is lying at the outer side and the bases are stacked in between. So, like this you can visualize like this, one chain is going up, another chain is going down, bases are linked in between, they are present inside and then they are further bonding with one another. Now the adjacent bases or nucleotides are separated by the same distance of 3.4 angstrom, which we call as helix rise, means one base has got a thickness of 3.4 to another 3.4 is added, then 3.4 is added. So like this the helix is rising and the helical structure repeats every 34 angstrom. It means for one complete turn there are approximately 10 bases in the helix. Another feature of Watson and Crick model is there is a rotation of 36 degrees per base, that is 360-degree full turn for 10 base pair takes place. Approximately it is said 10, actually it turns out to be 10.4. Now strong and straight hydrogen bonds between the specific bases, that is A and T and G and C, is taking place and the diameter of the helix is 2 nanometer or 20 angstrom, means the distance to the total distance between the two helix is 20 angstrom. Since so far we were talking about DNA as a storage molecule, although we talked about the structural components of both DNA and RNA. So what is the significance of RNA? Unlike double-stranded DNA, RNA is generally single-stranded molecule with much shorter chain of nucleotides, and it is double-stranded only in the case of tRNA or transfer RNA, but generally non-helical in structure. The self-complementary sequences in biologically active RNA allow some of its part to fold and pair to form short helical loops, which is termed as hairpin loops. Now what is self-complementarity? Self-complementarity is, we talked about the two helices running in anti-parallel orientation, but this helix is capable of pairing with another helix, means if here at nth position I have got adenine, then in another helix at the same position we have got T, so that AT base pairing can take place. This complementary base pairing is termed as self-complementary, and the two helices in DNA are self-complementary to each other, so that they are able to base pair. So far we talked about the structure, or you can say the components of nucleic acid which form the structure of DNA. Now we will go back to the historical lens and see how the discovery of DNA took place. This journey of discovery of DNA, the determination of its structure and the recognition of DNA as a genetic material has been long and very exciting. Number of questions were raised, and to answer these questions, number of techniques were invented at that point of time. We know Watson used X-ray diffraction data, structure of bases and Chargaff's finding. The structure of bases were known long back in 1880s, proposed by Emil Fischer. So how X-ray diffraction data and Chargaff's finding helped Watson and Crick to elucidate this structure? Let's see at it. Erwin Chargaff and his colleagues in late 1940s found that 4 nucleotide bases of DNA occur in different ratios in the DNA of different organisms, and that the amount of certain bases are closely related. These data collected from DNA of many species led Chargaff to following conclusions. Base composition of DNA generally vary from one species to another. DNA specimens isolated from different tissues of same species have same base composition. Then the base composition of DNA in a given species does not change with organism's age. He also found that A always pairs with T and G always pairs with C, and reached to the conclusion that the sum of purine residues equals the sum of pyrimidine residues. This relationship is sometimes termed as Chargaff's rule, and this was a key to establishing the three-dimensional structure of DNA. How Rosalind contributed to this discovery? She was working on X-ray diffraction technology. In 1951, when she joined King's College London, she applied the X-ray diffraction methods to the study of DNA. She found that the density of DNA, and more importantly, established that the two molecules existed in a helical conformation. And by the end of 1952, when Watson and Crick met Chargaff, Chargaff shared his findings with the Watson and Crick, and by that time Rosalind also contribution was known. So this all together led to the foundation for James Watson and Francis Crick to suggest that the structure of DNA is a double helical polymer, and the spiral consists of two strands of DNA wound around each other on the 28th of February 1953. Here you can see the rod and metal structure of DNA model prepared by Watson and Crick. Now to sum up, we can say that the structures of both DNA and RNA make them so well suited that their major biological roles of storing and genetic information becomes possible, and this information is fundamental to the growth and survival of cells and organism because it specifies the structure of molecules that make up a cell. And the secret of this structure lies in the structure of nitrogenous bases, base pairing, and the three-dimensional helical arrangement. Thank you.